Hello, in this video we're going to talk about the processes involved in meiosis. So let's not waste any of your time. Let's get right to it. Now when we're talking meiosis, one of the first things we want to remind about is that it has two rounds of division. And just like mitosis, you got that PMAT, prophase, prometaphase, metaphase, anaphase, telophase. They're all there, although the one that, when looking at the diagrams, won't be discussed much. It's certainly there, but it doesn't have its own action as far as events that warrant a slide or a particular video portion. But the two rounds, meiosis 1, meiosis 2, again, all the phases are there. A couple of things that you're going to be introduced to when we talk about meiosis 1, and we mentioned in the previous video, is that these ideas of genetic variation and things that meiosis does that mitosis does not do in order to create that variation are things like crossing over, independent assortment, and we'll talk about those. But the idea of crossing over, in other words, exchanging genetic information, and we mentioned that as one of the benefits of sexual reproduction life cycle, is the fact that you can kind of fix any negative chromosomes or negative aspect chromosomes or things that can cause conditions that are not favorable for the offspring. But during prophase 1, some things are set into place to where this crossing over occurs. The idea of the two chromosomes being attached by cohesion proteins to develop a synaptotemal complex. When they're joined together, you can also see this referred to as a tetrad or bivalent, as you can see there. But you'll see a picture of it here on the next slide. But it's just the idea that the two homologous chromosomes, one from the mom, one from the dad, are literally joined together, so this crossing over process can occur. Now here's a picture. Again, blue the dad, if so, red the mom, or vice versa, whichever you prefer. But just a recognition that one's the dad, one's the mom, and you can see this kind of binding to each other. Not only are they connected at the centrosome and the kinetocheres, but you can see the little spikes, the little cohesion proteins that kind of hold them together, so this process of crossing over can occur. And you'll see during meiosis 1 video, we'll talk about crossing over more directly. Alright, but again, we want to emphasize features and processes of meiosis. The idea of these the extra set of divisions, the reduction division in meiosis 1, it deals with the homologous chromosomes. You've got the chromosomes from the dad, chromosomes from the mom, and the homologous ones, the ones that are featuring the same traits, whether it be hair or height or skin color, whatever it may be. But it's also a reference to potential test questions and quiz questions. When the question refers to homologous chromosomes or homologous pairs, we're looking at meiosis 1. When you see a reference to separating sister chromatids, or the questions about sister chromatids, we're talking about meiosis 2. Now, so in reference to the cell cycle, you've got your typical cell cycle. You go through interphase G1, S, G2, and then you go into meiosis 1. But in between there, there is no further DNA replication. I mean, you don't want to separate them and then make more. Excuse me, make more. Then you've got to go through the process again. So no duplication, which infers that in between there's no S phase, and we'll talk about that. We'll touch on that again. You do go through the checkpoints, just to make sure everything's going well. We certainly want to do that. And when we get to errors, we'll talk about ways in which, when it doesn't go well, what can happen. But the idea of separating the sister chromatids in meiosis 2, and then you get down to those 4. So from one cell, you get down to 4 haploid gamete cells, which can be sperm or egg, depending on what the gender is. All right, so again, this is the same diagram we saw a few minutes ago. It's the idea of getting that first round of division where you separate those homologous chromosomes, and then in round 2, meiosis 2, you're separating the sister chromatids. That's the huge take-home message for this diagram. And then the cells that go through meiosis, as I mentioned, they're gametes, but they're germline cells. Okay, and then those germline cells eventually produce the sex cells, or the gametes. Well, that's our video for the processes involved with meiosis. When we come back next time, we're going to be looking at meiosis 1. We'll see you then.